Hello everybody, and welcome back to my YouTube channel. And in today's video, we will be learning about certain MCQs of hospital and clinical pharmacy, and also certain parts of process validation and quality assurance. So, when I talk about process validation and quality assurance, quality assurance, there could be questions on testing for packaging materials, the different analytical validation parameters like accuracy, precision, LOD, LOQ, right? Robustness, ruggedness, rigidness, intermediate precision. So all the analytical validation parameters, then the types of qualification, IQ, OQ, DQ, PQ, the difference between them, like design qualification, process qualification, and all. So operational qualifications, they could be asking you upon questions upon that. Then ICH guidelines, because ICH has a lot of guidelines on quality management, risk management, and all. So that could be one of the possible questions. So process validation is a documented program that provides a high degree of assurance in a process that will consistently produce a product meeting for predetermined specifications. Predetermined specifications. So here the right answer has to be Q7. That is the quality management system, or Q8 is quality management system. Yes, Q8 is quality management system. Is it? Let me know in the comment section that which of the following ICH guideline is for quality management system, which one is for pharmaceutical development system, and which is for risk management system. And is Q7 for good related to good manufacturing practices and high degree of assurance because of predetermined specification? Let me know. So the right answer becomes A, the CCP. So when we talk about quality assurance, you need to know certain things about QBD and DOE. There are certain tools like QTPP, Quality Target Product Profile, CPP, Critical Process Parameters, CCP, critical control points. So you need to know that there are certain points and parameters on which your entire quality assurance could be based upon, or design of experiments. Like in what are the input output variables, what are the levels, what are the factors, two factorial, three factorial, all those things. Because in an experimental design, there are a lot of different confounding variables or experimental variables that come into picture. So it is taken into account by the DOE experimentation. So you can do that as well and learn about that as well. Then CQA, critical quality attributes, CMA, critical material attributes. So all these are very very important specifications that you need, or the points that you need to know. So the critical control point is basically a point where hazard can be prevented or reduced to an acceptable level. Moving on, we have certain questions from pharmaceutical calculations. So power number. So power number is basically useful in mechanical mixing. So mechanical mixing is the right answer, option number D. The Reynolds number is, obviously I've mentioned this in my previous session, that it is less than 2000. Particle size distribution. When we talk about particle size, obviously it comes to sieving: coarse, moderately fine, fine, and very fine. There are different grades of courses of powders. So the right answer is sieving. DSC is for if you want to determine any interactive studies like DSC, DTA, differential thermal analysis, thermogravimetry analysis for moisture content or certain amount of hygroscopicity, FTIR and all for drug polymer interaction you can do. For protein interaction, you can do circular dichromism or XRD and all, right? For crystalline substance, XRD. So please make sure that you are aware of the techniques and in which what are the applications. In tablet manufacturing, which process is used to apply a uniform layer on the surface of tablets? So the over here, the right answer is when you talk about uniform layering. So it is film coating, or it can be sugar coating, or it can be enteric coating. So it has to be coating. Lyophilization is basically freeze drying, something that is done by freeze drying. So the right answer is drying by freezing. Which equipment is used for sterilizing pharmaceutical products using moist heat? So using moist heat, basically you use a autoclave, 121 degrees Celsius for 15 minutes for 15 psi, right? And steam under pressure, it's also known as the steam under pressure. Fluidized bed dryer operates on the principle of convection. It operates on the principle of convection, where there is actual movement of particles. In conduction, there is no actual movement or migration or transfer of particles. But in convection, there is an actual transfer of particles. Then which of the following is a method of sterilization for heat sensitive? Obviously, then heat sensitive can be sterilized by filtration. Which of the following is used to produce a fine powder? So if your fluid energy mill was there, that would have been the right answer. But here another synonym of fluid energy mill is there, which is micronizer pill or jet mill. So please make sure that you know this ball mill is not used for fine powders. Hammer mill is also not used, but jet mill is definitely used for fine powders. Good manufacturing practices. Then similarly, we have good laboratory practices, good documentation practices, GCP, good clinical practices, GWP, good warehousing practices. There are different kinds of practices that you need to perform. GAP, good archiving practices. So there are certain SOPs, standard operating procedures. And also while you're in the pharmaceutical industry, where you're keeping a data, so there are BMR, batch manufacturing record. Then there is master formula record, MFR. So you need to know about the BMR, MFR, the components, batch number, weight, and all, right? Manufacturing date, expiring date, which is used to determine the hardness. Hardness can be determined by hardness tester, while abrasiveness can be determined by friability. A sieve shaker is used to separate particles based upon the size. Which of the following method is based upon the drying of heat sensitive pharmaceuticals? So, heat sensitive pharmaceuticals are basically freeze dried. You freeze dry them because they're heat sensitive, so you cannot apply any heat source. So, spray drying method you cannot apply only because there's heat, right? Well, what is the main purpose of fluidized bed granulator? The fluidized bed granulator is used to dry as well as the coat. So drying as well as coating, but sometimes coating is a additional thing. Main purpose is for drying. So the right answer has to be dry the granules. Which one of the following processes is used to improve the flow properties of powder? Flow properties of powder can be improved by promoting the granulation process. Dry granulation, wet granulation, direct compression, chilsonator or slugging. Right? These are different techniques. Which dryer is most for drying granules and powders in pharmaceutical industry? Obviously, it's FBD. Compression. There are different terms: compression, compaction, and consolidation. Compression, compaction, and consolidation. So compression is basically a formation of a tablets from powders. You're compressing powders in a die to form a tablet with the help of punches, upper punch and lower punch, right? So that the cam track guides the movement, and whenever there is a feed of the powder from the hopper, it appropriately gets into the die, gets punched, moves to the station, and then finally comes out as a tablet. Which equipment is used for blending powders in pharmaceutical manufacturing? Again, it is V blenders, double cone blenders. V blenders are most commonly used mixers in pharmaceutical industry. The process of converting a liquid into spray and drying it rapidly is called as spray drying. Then in the spray drying, there is quickly conversion of a liquid into fine droplets or fine spray particles. Tablet manufacturing disintegrant is any substance that is used for the tablets to break down into small fragments. So there can be super disintegrants like sodium starch glycolate, cos carmelo, so there can be disintegrants like starch. So disintegrants can be having different mechanisms, wicking, swelling, all these are mechanisms of disintegration. Which process is used to increase the surface area of a solid? So the in the process of increasing surface area, we reduce the particle size, which is called as milling, pulverization, milling. Right? These are all synonyms for particle size reduction. Friability is basically a term in which you determine the resistance of tablet to break, not the hardness of a tablet. For that, we have Monsanto Verveca hardness test. The resistance to crumbling, the resistance for abrasiveness. The primary function of autoclaving is to obviously sterilize equipments. Next, we have critical quality attributes in tablet manufacturing. Critical quality attributes are something that are not organoleptic or something. They can be dependent, like on the related to the quality. So it is hardness. Hardness is a kind of a CQA, critical quality attributes. You can even quantify the hardness that it is this percent hard. USP, United States Pharmacopoeia. Which equipment is used to measure the uniformity of liquid products? So Silverson emulsifier or Silverson homogenizer is basically used for emulsion products as well. Next, we have certain MCQs from hospital and clinical pharmacy, and we will be covering in this lecture itself. Which regulatory agency is used for drug approval in United States? The right answer is MHRA. MHRA. Okay. United States, I'm so sorry, it is a wrongly marked answer. In US, you have US FDA, and in UK, you have Ministry of Health and Regulatory Agencies. This is for UK. TJ is for Australia. EMEA is for Europe. Anvisa is for Brazil. NAFDAC is for Nigeria. Similarly, we have MPA for Sweden, Medical Project Agencies. So you need to know about all of this. Then PMDA for Japan or MHLW for Japan. You need to know them very well. The main purpose of certificate of analysis, it's to confirm the quality of product. Whenever we talk about certificate of analysis, you usually whenever you order a bulk product or order an API, there is a certificate of analysis to refer to confirm the quality. In the context of Q9 is basically risk management. Q8 is pharmaceutical development, and Q8 is pharmaceutical quality system, and Q9 is pharmaceutical Q10 is pharmaceutical development. ISO 9000 is for quality management, while ISO 14000 is for environment management, EMS and QMS. Please remember the difference between them. Which one of the following guideline is controlled with stability testing of new drug substances? When I talk about new drug substances, it is Q1A. Q5 and Q6 are for biotechnological products and pharmaceutical products specifications. So the right answer has to be Q1A. Q1B is for photostability studies. Q1D is for bracketing and metrics. And Q1E is for evaluation of stability data. Right, so you need to know about all of this. New drug substance is Q1A. New dosage forms or something is Q1C. CAPA. Corrective action, preventive action. Option A is the right answer. Type A is usually dose dependent and predictable reactions, while type B is dose independent or idiosyncratic and unpredictable infection. Type 1 hypersensitivity is immune mediated, and while type 4 is the cell mediated in immunity. Hypersensitivity. Delayed hypersensitivity is basically type 4, where serum sickness and where tuberculin skin test is one of the examples. Pharmacovigilance is basically a science on activities leading to detection, assessment, evaluation, and understanding and prevention of adverse drug reactions. What is drug utilization review? When I talk about drug utilization review, it is measure of evaluation of appropriateness. One MCQ from my side to y'all. In the pharmacy and therapeutic committee, who is the chairman and who is the secretary? Let me know how many people can know this. In the pharmacy and therapeutic committee, who is the chairman and who is the secretary? Pharmacist is what, and medical practitioner is what, like doctors are what. Okay, let me know how many people can know this answer. Which of the following is not a chemopharmacotherapy plan? So pharmacotherapy plans are basically very important plans, and we usually study them in hospital and clinical pharmacy. But sometimes in the pharma syllabus we don't have them. Matlab, usually in the B Pharm syllabus you don't have these kind of pharmacotherapeutic plans. So you should know that the soap pharmacotherapeutics are very very important. Prime pharmacotherapeutics is very important. Right? Soap is subjective, objective, assessment and plan. Subjective, objective, assessment and plan. That is your soap pharmaceutical analysis. When I talk about prime pharma therapeutics, prime is also one of the scheme which is run by Europe. So prime pharmacotherapeutics is very pharmacotherapeutic plan, basically pivotal, identifying and all. So you should be knowing the different pharmacotherapy plans. So the answer is none of the above, because they all are pharmacotherapeutic plans. Core, farm, soap, they all are pharmacotherapeutic plans. So that's the right answer. So that's it for this series of pharmaceutical MCQs top 25. I hope you all enjoyed it. Feel free to drop your doubts in the comment section. Let me know if you all want any more series to come up. Thank you so much for watching the video.